Hi everyone, welcome back to the channel. I'm the GCSE science teacher, and in today's video we're going to be talking about a required practical for GCSE biology students. So if you do enjoy the video, feel free to give it a like, share it with someone else you think would benefit from learning this, and also do subscribe so you don't miss out on any other future uploads that I have. I do make videos for chemistry and physics GCSE as well, so if you are interested, have a look on the channel. You'll see lots of playlists of videos that I've already created for your GCSE revision. And if you have any video suggestions, feel free to comment them down below as well. So let's get started. Let's talk about photosynthesis as a bit of a recap. I did do a video on this and I'll link it in the cards for you if you want a refresher. But just in short, remember photosynthesis is an endothermic reaction. Light energy is absorbed during that reaction by chlorophyll, which is found in the chloroplasts of plant cells. Not all plant cells have chloroplasts. Remember, root hair cells don't, but any of the other plant cells will have these to allow photosynthesis to occur. Usually we're talking about the palisade cell. And photosynthesis is very much dependent on the temperature of the environment, but also the carbon dioxide concentration and light intensity. Remember, light intensity is just how bright that light is. And although, yes, a high temperature, high light intensity and high carbon dioxide concentration is often beneficial for photosynthesis, if you have too much of an extreme of anything, it can actually be a negative. So we're going to talk about this in terms of graphical data in a moment as well. In terms of this particular practical, it's mainly focusing on how light intensity affects the rate of photosynthesis. But you could also investigate how the other two factors affect the rate as well. Something also to be aware of in this particular practical, you could be expected to write a method for it. You could be expected to interpret data for it as well. So that's just something to be aware of. So these are the limiting factor graphs that are to do with photosynthesis. And remember, there are other factors that can affect how photosynthesis occurs and the rate at which it occurs. By the way, the word rate is just talking about how quickly something happens in a given or set amount of time. So another factor that's not displayed here is how much chlorophyll there is. This will obviously depend on the species of the plant. It will also depend on how the plant is situated in the environment and whether there are any diseases associated with that plant. So, for example, TMV or tobacco mosaic virus, there is something going on there called chlorosis, where there is less chlorophyll present because of the viral disease. And if you want more information about plant diseases and just diseases in general for GCSE, I did do a video on this and I'll link it for you. So let's have a look at these graphs. We've got temperature, carbon dioxide, and also light intensity. The light intensity graph and the carbon dioxide concentration graph will look very similar. There is like a nice linear line. You have a plateau at a certain point. However, temperature kind of goes upwards in a peak and back down. So let's kind of interpret these graphs and know what's really going on. So with temperature, the rate of reaction or the rate at which photosynthesis happens at the beginning, as temperature starts to increase, we also have an increase in the rate of photosynthesis. And this is because those particles of carbon dioxide and also water are given some energy to move around and collide to form glucose and oxygen. Also, temperature will affect the enzymes within the plant cell itself, and those enzymes are catalysts for various reactions, and this will help with the process of this sort of reaction as well. However, there comes a point where we get a optimal temperature for that plant and photosynthesis. So at this particular point, this is the highest peak of the graph. This is the perfect, optimal, the best temperature at which photosynthesis will happen. Anything higher than this temperature, enzymes will actually start to denature and the plant will no longer be able to photosynthesize. And this is a problem, because actually that plant, it's gotten to the point where there's no more photosynthesis that can happen, but also the plant starts to get damaged, and this therefore starts to reduce the rate into the point where it then just does not photosynthesize at all. So that's what's going on with that graph. In terms of the carbon dioxide concentration graph, we need to look at it in a different way. So again, as the concentration of carbon dioxide increases, the rate of reaction increases too. And this happens at a point where there is a plateau. So the plateau indicates that the rate of reaction is just going to maintain the same. It's not going to get any better. It's not going to get any faster. Even if we keep giving the plant lots of CO2, lots of carbon dioxide, there's no more photosynthesis that can happen. And this is because there is something else that is limiting the rate of photosynthesis. And that something else could be the temperature, or it could be how much light intensity there is. The same can be said for the light intensity graph. So again, as light intensity increases, so does the rate of photosynthesis until a fixed point. There is only so much light that can be absorbed by the amount of chlorophyll present. So even if we whacked on the light really, really high, the intensity actually makes no difference at this point. There is a plateau, and the constant rate remains the same. And that's because something else is now limiting the rate of photosynthesis. Again, it could be carbon dioxide concentration or temperature. So adjusting those different factors will then increase the rate further. So one thing you could get asked, like I said, interpret these graphs which we've just done together, but also plotting data as well, and possibly even predicting data from gradients and things like that. So just be aware that this is the sort of thing they could ask you in the exam. So during the required practical, your aim of the experiment is to investigate the effect of light intensity on the rate of photosynthesis. And to do this, you need to maintain the temperature and also the carbon dioxide concentration throughout. And these are what we call control variables. They are maintained and constant within the experiment. And this is to make sure that they have no effect on the data that we collect. And therefore, the data that we collect is reliable, but also can be comparable to other data sets of the same experiment, especially with things like peer reviews that go on in science. This is really important, an important step. So the sorts of things you're going to need, the apparatus that you'll need for this experiment include pondweed. You need to make sure it's the same type of pondweed throughout the experiment. There are loads of different species. I'll leave some in the description box of which ones they like to mention at GCSE. You also need to make sure that you have the pondweed in a solution of dilute sodium hydrogen carbonate. And this is a solution that contains carbon dioxide, so that you can make sure that the plant is getting a constant supply of carbon dioxide as it is submerged in water. And the reason it's submerged in water is so that you can count the number of bubbles of oxygen gas produced. You also are going to need an LED light bulb, and this is not a typical light bulb. It's an LED for a reason, because they are very efficient and they don't actually transfer or emit a lot of heat energy that could alter and affect our results. So this LED light bulb will maintain the temperature and make sure that there are no fluctuations in temperature as well. You'll also need to make sure you have a meter ruler or something to measure, like a measuring tape, so you can measure the distance as you do this experiment. Another thing, of course, you need to make sure you're writing it in a table so that you have your results nice and clear, so you can do some analysis afterwards. So this is the required practical method that you need to know at GCSE. One thing to say with any method at GCSE, or just in general if you do your A-levels as well, you want to make sure that the method you're writing is really easily conveyed to the reader, and that if they picked up your method and actually were to do the method and do this in a lab, they'd be able to obtain valid results. That's what will get you the biggest marks in the exam, and this is what we call a leveled response. So those people who get the highest marks will have numbered bullet points so that it's in chronological order. The steps are really clear and understandable to the reader. You've mentioned the key apparatus that you need throughout the experiment. You've also mentioned control variables, independent variables, the dependent variables as well, and potentially any hazards associated, especially if they ask you for that. That's really important you include it. This way, the reader knows that you actually have a method that will give them the best results, and they'd actually be able to interpret it and repeat this. Because in science, just as a topic, science is all about, can someone else do this? Can someone else repeat my experiment? And in the research world, that's really important. We use something called peer reviews, where other academics or the scientists or the researchers will use other people's information data from an experiment, and they will try to replicate it in their own lab. So it's really important that any method you write is really easily interpreted and easy to do themselves. So let's talk about this practical. Let's actually think about how we'd write this method. So you first want to get your pondweed, and you want to cut about 3 inches of the pondweed and place it into a beaker or a boiling tube, and this will contain a solution of sodium hydrogen carbonate. Like I said, this particular substance will provide the pondweed with carbon dioxide at a set concentration and amount, or a volume, as well as concentration. And this is a control variable, so the pondweed is maintained in that concentration of carbon dioxide, and that's not going to affect the rate at all because it's constant. You then want to just snip the end of that pondweed with some scissors, and this needs to be under the water. So the pondweed is submerged in the water and you snip off the end of it so that the stem, when it produces those oxygen gas bubbles, it's easily released and easily identifiable when you are counting for them in the experiment. So once you've done this, you want to just leave the pondweed in that water submerged for a few minutes. About 5 minutes should be fine, just to give it a moment to get used to its environment. And we call this that it's actually acclimatizing to its environment. After that, you want to place your boiling tube or your beaker of the solution with the pondweed in, and you want to place it around 10 centimeters away from the light source. Now, the light source itself will need to be an LED, as I mentioned. We don't want temperature to affect the rate of photosynthesis here, so an LED light bulb is used for that reason. Now, once you've given the plant a few minutes to acclimatize, you then want to measure or count the number of bubbles of oxygen gas produced every minute. And you want to record that reading in a table. You then want to repeat this so you get a few different readings of the same distance from the light source. And then you want to move your pondweed away from the light source by the same set increments. So let's say we start at 10 centimeters, we want to move the pondweed another 10, so 20, and then another 10, so 30, 40, 50, 60, etc. until really good range of results. And this way, once you've got all your recordings, you'll be able to see how increasing the distance from the light source, how it essentially affects the rate of photosynthesis. So what you should see is the further away the pondweed is, the less light intensity there is, and therefore there are less oxygen gas bubbles produced every minute. So the rate is actually reduced as light intensity decreases. Now, you may even find there is a plateau stage where the bubbles will stay the same, and this is because that tells you the optimum light intensity for that particular piece of pondweed. So it will kind of look, if we were to graphically display this data, it will kind of look like the graph you saw a few moments ago, and that's kind of the data you should get and the results you should expect to find as well. So I hope you did enjoy the video. If you did, please give it a thumbs up, share it with someone else, and do subscribe if you haven't already. I've been the GCSE science teacher and you have been curious. Thank you so much for your time today. If you'd like more videos or revision content, do check out the playlist I've linked, and also the other places you can find my content is on Instagram and TikTok. All of it is at the GCSE science teacher. In the meantime, have a great day and I'll catch you in the next one.